Okay, wonderful. Thanks a lot, Katie, for this very kind introduction. Indeed, it is a homecoming. When I was an undergrad, this was actually a prison standing here. Probably some of you might know. Um, and thanks a lot for the invitation to come here. It's really an honor to speak today. So the seminar was entitled also Microbiome for Personalized Medical Approaches. And one common problem we do have in medical sciences is the fact that if we have one cohort of patients that all have exactly the same disease and we give them exactly the same medication, individual might respond very differently to that drug. So we have the good responders, we have the people who don't even feel that they have taken a drug, and then there are some patients who might feel different side or adverse effects. The problem is we do not know a priori to which cohort a given person belongs to, which makes it really challenging to find the right drug for the right person in the right dose. And the way this has been tackled over the last few decades is actually looking at the human genes, about 23,000, to look for variations that explain and potentially predict drug response. But as we have already heard by Katie at the beginning, we were asking, what about the 2 million to 3 million gut microbial genes that we carry around with us? So indeed, we're all only about 1% human. So do the 99% matter for drug response? And also, this was pointed out, the fact that actually between individuals, our human genome might vary less than 1 or 0.1%, whereas between our microbiota, there might be differences that are as strong as up to 80%. Really giving the possibility that those differences might matter for interpersonal differences in drug response. And we looked mainly at drug metabolism, and we were asking as a first question, well, how can we even separate microbiota drug metabolism from host drug metabolism? Because if you take a drug is typically, as mentioned before, for nutrients, typically absorbed in the upper intestine, reaches circulation, hopefully has a beneficial effect until the drug is metabolized and eventually eliminated. However, drugs are typically not fully absorbed, and certain proportion or certain portions propagate in the gut where they see increasing numbers of microbes that could also metabolize the drug, and converted to metabolites that might also be taken up and have an effect on the system. So to further investigate this, we first look actually at the nucleoside analog. This is an antiviral drug because it had been described already in the 90s that it can get metabolized by both microbial and human enzyme to bromovinyl uracil, or BVU in short, which is important because this is a toxic metabolite because it inhibits DPD, which is the rate-limiting step for pyrimidine metabolism, or pyrimidine analogs such as 5-FU. And before this interaction had been recognized, it killed 18 people in clinics. So we were first asking, well, is indeed in our hands, do mammalian and microbial enzymes convert the drug into its toxic metabolite? And what you can see here is mouse and human liver extracts, as well as their microbiota, incubated with the drug, and then we measure over time the production of the toxic metabolite. And as you can easily spot from those assays, or almost stoichiometrical conversion, indeed, microbial and human or mammalian enzymes can convert brividine to BVU. Well, the question is, does this really matter in vivo? Because this is all in a test tube. And we then turn to germ-free animals. This is a picture from the lab where we keep animals that are completely germ-free. And Andrew has pointed towards germ-free animals, and actually Randy as well. So they live in here, so they don't have a single microbe living in or on their body, and they're a good control of microbiota negative animals. And then we took germ-free animals and conventional animals, and we did pharmacokinetics. We gave both cohorts of animals an oral dose of brividine, and then we followed in 9 different body compartments the drug and its metabolites. And I show here the lower intestine. And as we were expecting from our in vitro results, you can see that in germ-free animals, not having a gut microbiota, actually the drug accumulates over time, whereas in conventional animals, the microbiota converts the drug into its metabolite. However, interestingly, we do not see an according accumulation of the toxic metabolite in those compartments, suggesting that the toxic metabolite is taken up systemically. And indeed, when we look at serum, you see a microbiome-dependent accumulation of the toxic metabolite, clearly pointing out that the microbiota contributes to systemic BVU. And it raised the question, well, can we contribute or can we quantify how much contribution is there from the microbiota? And what we did, we set up a pharmacokinetic model, and I just show here in this example here how this works. So in this example, I described the kinetics of brividine in serum, which depends on the absorption of the drug minus the metabolism of the drug minus the elimination. And you can now set up, keeping the mass balance constant, you can set up a system of equation for all the different transitions. And as we have animals that do not have any microbiota contribution, we can actually parameterize the host as a model. What we then do, we keep the host parameters fixed, and we add the data from the microbiota colonized animals, and we ask the model, if we now add the data from the model with the microbiota, how much is the contribution to the toxic metabolite in serum from the host, shown in black, and how much is the contribution by the microbiota, shown in red? And you would expect, if you simply sum up those two curves, that this actually reflects the experimental data of the animals with a microbiota, which fits fairly well. And it allows us furthermore to actually quantify that 70% of the contribution of the toxic metabolite in serum is indeed directly through the production by the microbiota. So what I wanted to show you in this first part is, how do we dissect host and microbial drug metabolism using those bubble animals, how we do in silico modeling of the biological processes involved to really get a quantitative understanding of the contribution of the microbiota to drug metabolism. And probably most importantly, I wanted to show you there are cases in which microbiome drug metabolism matters for pharmacokinetics and drug toxicity, really bringing me to the second part of this presentation, where for brividine, I mentioned that there were already hints that the microbiota contributes to drug metabolism. However, for most drugs out on the market, we have no idea whether the microbiota can metabolize the drug. So how can we systematically do that? And does it help us to actually explain the metabolism of entire gut communities? So to get a first inventory of drug metabolism by gut microbes, we took a set of 271 clinical drugs, FDA-approved drugs, and we incubated them with 76 bacterial species isolated from the human gut. And then we used high-throughput LC-MS analytics to test which drug gets metabolized by which bacteria. Strikingly, we found that 2/3 of the drugs were metabolized by at least one of the bacteria we tested, which you can see here in this heat map, where you see again in the y-axis the 76 species tested, and in the x-axis the 176 drugs that we find metabolized. And each square is a drug bacteria interaction, and the brighter the square, the stronger the drug gets metabolized by the bacteria. When we then take this drug metabolic data and we perform some clustering along the species, you can see color-coded that bacteria actually, that we fairly well reproduce the phylogenetic proximity of those bacteria, which is good news. That tells us that the genetic content, the genetic information of those bacteria tells us something about their metabolic activity. When we then do clustering along the drug axis, according to how likely or how those drugs get metabolized, we cluster drugs together that share chemical units. For example, highlighted here, these are drugs that are mainly metabolized by Bacteroidetes species, and all those drugs have actually an ester group highlighted in red, suggesting that the bacteria probably hydrolyzed this drug. However, just looking at the parent drug doesn't really tell us what the bacteria do biochemically, raising the question, can we identify the biotransformation products? And to do so, we went back to our samples and we acquired untargeted metabolomics, detecting about 6,000 metabolic features for each sample. And I show you here one example, which is Bacteroidetes tetraioda omicron, incubated with the calcium channel blocker diltiazem. And you can see 6,000 gray dots representing 6,000 different metabolic features, but only 2 were actually specifically present in the condition shown here, incubation of diltiazem with B theta. One is actually diltiazem itself. That's a good positive control because that's what we spiked in. And the other one is actually a compound that is exactly minus 42.011 Dalton smaller, suggesting the deacetylation of that drug. We then performed this type of analysis for all the drug bacteria pairs and calculated the exact mass difference between the putative metabolites and the parent drug, shown here in this histogram. And what you can appreciate is that distinct mass shifts occur multiple times, suggesting that multiple drugs undergo the same chemical modification by the bacteria. And indeed, when we look again at our minus 42, which is a deacetylation, we have 9 drug hits, and all those hits actually have an acetyl group, or at least one, shown in red, that the bacteria can clip off. So as we now understood a little bit more about which bacteria metabolize which drug, and we got some insights in the underlying chemistry, we were wondering, can we identify the metabolic pathways, the enzymes responsible for those reactions? And to do so, we returned to our initial pipeline, but instead of testing different bacteria, we made actually a genetic library, expression library, where we took, in this case, Bacteroidetes tetraioda omicron. We took random pieces of the genome, cloned it into an expression library, arrayed it, and tested up to 50,000 clones by LCMS to identify clones that have gained the capability of the gut bacteria to convert the drug into an identified drug metabolite. So again, going back to diltiazem, after having screened 50,000 clones, we found 4 hits. And when we sequenced them, all those random fragments map back to the same locus in the B theta genome, covering 4 genes, but really actually all of them just covered 1, suggesting that BT4096 is responsible and sufficient for diltiazem metabolism, which you can see here in a biochemical assay. We could also reproduce it by really knocking out the gene shown here in black and bringing it back as a dashed line in a complementation assay, allowing us now, having a single knockout strain, to build a mouse model that is identical in both cohorts with the difference of that one single gene, and then do pharmacokinetics. And as you can see in those plots here, indeed we change the kinetics. We have more of the parent drug in dependent on that one single gene compared to the wild type colonized animal, and for the metabolite it's exactly inverse. So this is all on a single monocolonized, but I started at the beginning that we have so many genes, we have communities in our gut. Does this even help us? And that was the question we addressed here as a last point, where we took 28 human gut communities. We cultured them under anaerobic conditions, and we incubated it with diltiazem to measure the conversion to its metabolite, shown here in the middle, where each color shows the data of a different gut microbiota. So the first thing you should notice here is that indeed there's large interpersonal differences in how those persons' microbiota metabolize the drug. And then because we had the gene in our hand, we could design a qPCR assay to quantify the abundance of the gene. And actually, quite surprising, there's a fairly good correlation, because chemically speaking, a deacetylation is not such a crazy chemical or biochemical reaction. Nevertheless, it seems that in those communities of 2 to 3 million genes, there's one gene that fairly well explains the activity of the entire community. And that's something that Eleonora is further developing, making models also for different drugs to eventually predict those interpersonal differences. With this, I want to summarize my second part. I want to show you how we use high throughput analysis to systematically identify specific bacteria-drug interactions, how we then go about the identification of metabolic enzymes and pathway, and that once we have them in our hands, we can actually assess whether they would be good biomarkers for microbiota drug metabolism. So now I changed the field from drug. We actually got more and more interested in biotransformation of other organic molecules, really by the observation of a lot of cancers are triggered by the exposome or by chemicals in the environment humans might get exposed to. And the other observation is there's barely any cancer that has not been associated with any microbiome signatures. And we thought, well, can we bring those two together? And the way we did that is actually, the question we first asked is actually, does the microbiome contribute to the toxicity of those environmental carcinogens? And this is a collaboration with Blanc and Janos from Split University and Boyawe Eleonora and George from my lab. And the first thing we did, we actually took a well-established model for a carcinogen-induced bladder cancer, where you give BBN, which is a nitrosamine that is also found in cigarette smoke, in drinking water to mice over 12 weeks. And then after an additional 8 weeks, the mice typically develop bladder cancer. We added a second group where we depleted the gut microbiota, giving them antibiotics in parallel, and then we compared through histopathology the cancer development in the bladder, which you can see here from bright purple to dark purple, kind of increasing severity of bladder cancer. In each group, we had 30 plus animals, and you can easily see from those stacked bar plots that if you have a microbiome, so you just give the carcinogen, most of the animals develop cancer. However, if you in parallel also give antibiotics, the majority of the animals do not develop cancer, really suggesting that the microbiota contributes to cancer development. We were then first thinking, well, it's just a difference of the carcinogen in the bladder, which we actually didn't find to be true. However, when we looked at the metabolite of BBN, carboxy-BBN or BCPN, we actually did find a difference, and we did find significantly more of the metabolite in the group that also developed more cancer. Really raising the question of where is this BCPN produced? And to trace that, we first looked at host tissues, liver, kidney, and plasma, but we didn't find a significant difference. We also took hepatic cells, we exposed them to antibiotics to test the hypothesis whether antibiotic exposure would change the metabolism of the liver cells, which we also didn't find to be true until we actually started looking into intestine, and you see here duodenum all the way to rectum, so along the gut. We found in animals with a microbiota, shown in black, we found this huge accumulation of BCPN, which was absent in the presence of antibiotics. We then were wondering whether it's just a conversion of BBN, which we didn't find to be true. So what we actually found is that glucuronyl BBN, so the type phase 2 metabolite of BBN, is actually probably converted into BBN, which is further converted into BCPN. The question was, can actually the microbes do that? Because this is an oxidation reaction, and our general assumption is that the large intestine is mostly anaerobic. So we wanted to test that directly with our system. So you've seen this pipeline before. So this time we took BBN and we took different parts of the large intestine, cecum, colon, and rectum, and we incubated it under anaerobic, microaerobic, and aerobic conditions. And you can see here when we follow over time the production of BCPN that all those microbiota communities, they can actually produce BCPN as soon as oxygen is in the system. What we then, to identify the bacteria that can do it, we assembled a collection of 600 different bacteria, isolated under microaerobic and aerobic conditions, and we tested each of them for its capability to produce BBN to BCPN. And through sequencing, we identified 12 different species that in our hands could metabolize BBN to BCPN. To test whether they're actually also present in the gut microbiome, we did 16S analysis, and you can see that they are present, but they're fairly low abundant in the around 1% or even lower for most animals. We have those converting microbes around. So far so good for the animal model, but what about human? Does it actually matter, the mechanism of carcinogen metabolism by gut bacteria in human? And we performed 3 different experiments to test that. In the first one, we took 10 fecal samples from different healthy humans, and we, with our system, we tested whether those communities can convert BBN to BCPN. And indeed, you can see that some of those communities can very well convert BBN to BCPN under microaerobic and aerobic conditions. You can also appreciate from this plot that there seem to be quite interpersonal differences in how those communities can convert the carcinogen. The second experiment we performed, we took germ-free animals. We did a fecal transfer from G11, that community, into mice, and we performed the initial experiment with BBN and BBN antibiotics, and we could actually reproduce the phenotype that we also had with the mouse microbiota. And finally, we made a library of 500 microbes that we isolated under microaerobic and aerobic conditions, tested them all for their capability to convert BBN to BCPN, leading us to the identification of 11 different species from human that can actually metabolize BBN to BCPN. Which then raised the question, well, are those bacteria sufficient for the phenotype? And to do that, we did what also Randy pointed out. We did a monocolonization of germ-free animals and compared them to germ-free animals. And indeed, you can see having that one bacteria in the gut leads to the same accumulation of BCPN in the large intestine. Of course, a monocolonization is not fully reflecting what we have in human. Also, a 2- and 3-member community is not fully reflecting. However, that's the next step we took. We took a 2-member community and a 3-member community. We gave them both BBN and we followed intestinally BCPN and BBN. And you can see that the addition of that one metabolizing strain indeed leads to significant changes in the large intestine. So how general, and this is the last question I'm gonna pose for today, how general is carcinogen metabolism by gut bacteria? And Boyao, who was a PhD student in the lab, he took this further. She took that pipeline, but then she took 68 different organic carcinogens and 34 gut species. And the ones here in red are gut species inspired by what I showed you in vivo, gut species that can grow both microaerobically and also anaerobically. So the red ones, they're found in the upper intestine and the oral cavity. And she tested how each of those microbes metabolizes those 68 carcinogens. And I show you 2 heat maps. This is the microaerobic one, 5% oxygen. This is the anaerobic one. And again, the brighter a square, the stronger a given carcinogen is metabolized by a given bacteria. So she found there are differences under microaerobic and anaerobic conditions, which is not that surprising. The presence of oxygen changes metabolism and the biochemistry. And also, she found that in total, 48 of the tested 68 organic carcinogens can be metabolized by gut bacteria. And with this, I come to the summary of this part. I wanted to show you how the microbiota can biotransform nitrosamine carcinogens, which then impacts the toxicokinetics in vivo, and that gut microbial nitrosamine metabolism actually can promote bladder cancer through those alterations of the toxicokinetics. And in the last point, I just want to give you a snippet of how microbiota carcinogen metabolism is actually seems to be fairly general and could in the future be a risk factor for the predisposition risk of tumor development. We're currently trying to follow up on several of those metabolic reactions to ask, well, does gut bacterial metabolism of carcinogen, does this actually change the mutagenic potential of those carcinogens? Does it change the toxicokinetics? And how does it, for procarcinogens that need activation, what is the role of the microbiota for those? And with that, I want to thank my group. I think I pointed out people who have done the work, several collaborations really across the globe, and funding resources. I thank you for your attention, and happy to take questions in the Q&A session.